The calculations and measures of our study are based on predictions of exposure calculated at each study site. These predictions are directly affected by the human population, as the sewage effluents are directly proportionate to the density of humans in the area surrounding the test sites. Each test site will then be categorized as a high, medium, or low impact site. These categorizations are made based on the assumed additive potency of the estrogens and on empirical data gathered from previous experiments. An important factor recognized in helping our study's materials and methods is literature review. We'll be using previous studies as a guide to the predictions being used, as well as understanding VTG induction and intersexuality. Our statistical analysis will be focused on plasma VTG concentrations and the considerations affecting these concentrations, like the risk category of the test site, the sex of the fish studied, the age of the fish, and the time of sampling. The data are gathered and placed into a table to assist with identifying proportions and trends, as well as making calculations like chi-square. The calculated estrogen concentrations in the rivers where roaches were captured and tested ranged from 0.07 to 13.78 nanograms per liter for E1, 1/100th to 1.7 nanograms per liter of E2, and from 0 to 0.37 for the EE2. With the exception of the estrone, the concentrations were normally well within an acceptable level and not considered to be high enough to cause feminization in fish. But, as previously acknowledged, our results did show that the estrogens were additive in nature. The results of the biological analysis showed that intersexed fish were present at the majority of the sites sampled. When the fish were pooled together, clear differences were noted between the high, medium, and low risk groups for incidence of intersexuality and severity of the effects. However, when the VTG analyses were pooled together, there was no clear dose-dependent relationship between VTG and E2, equivalent in the male and intersexed fish. After review of the results, this observation is believed to exist due to the fact the fish were sampled at different times of the year, thus affecting the outcome of the results. Figure 1, as shown here, shows the proportions of males, females, and intersexed male fish within each risk categorization of high, medium, and low. Unfortunately, it was not statistically possible to compare the sex ratios for all of the individual sites because an insufficient number of fish were captured at some of the sites. Several sites showed a departure from the original 50-50 ratio desired. At some of the study sites, as shown by the chart, more males than females were captured. However, at most of the sites, more females were captured than males or intersex males. In the end, when all of the data was pooled together, no statistically significant difference was found between the proportion of females in each of the risk categories. After a histological examination of the testes of the male roaches, it was found that a significant proportion of these fish were intersex, or characterized by the development of eggs within the testes or an ovarian cavity in addition to the sperm duct. In general, the intersex fish were identified in the medium and high risk categories and in a much more limited frequency in the low risk category sites. It was determined that the overall prevalence of intersex fish were influenced greatly by the exposure of the fish to the concentrations of the estrogens and the severity of the concentrations they were exposed to. In addition to the severity and duration of exposure, the fish's ages also were shown to be a factor in intersexuality, where the eggs were present inside the testes. Oddly, age did not seem to be a factor with fish who had a development of an oviduct. According to Jobling and her colleagues, feminization of the reproductive ducts is known to occur during early life, whereas the incidence of ovotestes appears to be a progressive condition. By looking at the chart, you can see that males exposed to estrogens for a longer period of time are liable to show signs of feminization by the presence of eggs within the testes. The VTG induction came back with interesting results. First, the VTG analysis provided strong evidence that the wild roach populations were being exposed to estrogenic contaminants. However, the predictions of the VTG induction did not correlate with what was found from the biological analysis. Many of the sites where VTG induction was not predicted to show signs within the fish did. Conversely, sites where VTG induction was expected ended up showing no signs in the fish at all. Through our literature review, we found documentation that suggested long-term exposure results in a lowering of the threshold concentration required to invoke a VTG response. Other documentation stated that lifelong exposure to EE2 may show a reduced VTG response of males because of acclimation to the environment. In addition to these other factors, the VTG concentration showed seasonal variation, especially for the female roach. While it was somewhat expected in females, there was a surprisingly similar trend in the male and intersexed male fish. After understanding what leads to intersexuality in fish and the results of our study, this brings us to the discussion: what is the implication of our findings on humans? The first major implication is how lowered fish populations would be impacted by endocrine disruption. The obvious is that the roach is a sports fish, both consumed as food by humans and used as bait by sports fishermen. If the populations drastically decrease, the fish can be removed as an option for the sports fishermen and for those who are eating the roach as part of their primary diet. Like all aquatic environments, when the aquatic organisms are affected by either having a dip or spike in the population, the homogeneous nature of the ecosystem is affected. The roach eats insects, other small animals, and aquatic plants. If the roach populations decrease significantly, we could see an increase in insect populations or aquatic plants, opening up the waters to an imbalance that can affect everything that relies on the water for life. Our second discussion point related to the implication on humans is directly related to the thoughts that if estrogen contaminants are in water and affect fish, how can we be sure that humans are not also being physiologically affected in the same way? The truth is, we can't. Joel Blyfus released an article in 2008 that relates intersexuality, cancer, and early puberty back to the same EDCs that are causing intersexuality in fish. For example, in 2004, the Potomac River was noted as having a high instance of intersexed fish. The same river is the primary source of drinking water for the Washington, D.C. area. In 2003, the South Branch of the Potomac River in Hardy County first produced egg-bearing male fish. Consequently, in 2004, a survey of women in Hardy County showed a higher than normal rate of ovarian and uterine cancer. Nancy Holt, a retired nurse, has also done some non-scientific investigation of her own. Nancy was spending time researching the area around Maybane, North Carolina, after noticing a strange trend in neurological problems, high cancer rates, and increased risk of staph infection from playing in local creeks and lakes. In her research, she found a low puberty age with girls developing breasts around 5 or 6 years old and young boys getting armpit hair around the same age. She attributes everything to the treated sludge being used as fertilizer on surrounding farmland, sludge that inevitably houses endocrine-disrupting chemicals. With these discussion topics in mind, I bring this presentation to a close. However, after you walk away from here, I truly hope you continue to think about how EDCs are affecting our local water sources and how we may possibly be affecting human populations through more than just the effects EDCs have on fish.